Okay, so this is chapter 18, which is blood, which I think this is one of my favorite chapters. It might be my favorite. Okay, so hematology, which makes sense, right? Anything with heme is blood. So hematology is the study of blood. Now, what happens sometimes, well, what happens all the time is hematology in children is oncology, so the study of cancer and blood. And this is in kids, because most of the time childhood cancers are actually leukemias. So oncology and blood for pediatrics is under hematology. Okay, so basic functions of the circulatory system. So one makes sense, right? Transport and exchange. So we transport oxygen, nutrients, and hormones to the cells, and then we take waste away from the cells. Protection. So protection would be immune system. So white blood cells are responsible for inflammation. They're going to help prevent infection from spreading. They'll destroy microorganisms or cells that aren't self. They neutralize toxins, and they also will keep us from bleeding to death. Okay, so they initiate blood clotting. The third thing is regulation. So the blood is really important to regulate body temperature. It regulates the pH of your extracellular fluid, your ECF, and it's also going to keep your fluids in homeostasis. So three big, broad functions of blood. Okay, so now the components and general properties of blood. So in general, if blood has oxygen in it, it's bright red or scarlet. So we don't say oxygenated and deoxygenated because there's always some oxygen in your blood, but what we say oxygen rich or oxygen poor. So it's red if it has oxygen in it, and then if it's oxygen poor, it's kind of more purpley or dark red. Okay, the volume of blood that one person has is between 4 to 6 liters in adults. So females are on the low end of that range, so 4 to 5 liters, and males are 5 to 6 liters. Okay, blood is 4.5 to 5.5 times more viscous than water. So viscosity is like how thick something is. So think about like maple syrup and Karo syrup, how or even like motor oil. People talk about motor oil. So the thicker something is, the more viscous it is. Or you can think about a milkshake and orange juice. Okay, so the milkshake would be more viscous. Okay, the temperature of blood is 38 degrees Celsius or 100.4 Fahrenheit. So it's slightly above body temperature. And then the pH of blood, this will be important as we move on. The pH of blood is 7.35 to 7.45. So there's a very narrow range where it has to be for the proteins to function properly. Okay, so if you think back to histology, blood is a connective tissue. It has cells. The cells we call formed elements. Okay, so the cells are formed elements, and then the extracellular matrix is going to be plasma. Okay, so the components of blood, so if you did a blood draw on someone and you put it into a tube, okay, and you put it in a centrifuge and spun it down, okay, or you let it sit and let the heavier things settle out, what you're going to see is red blood cells, which are also known as erythrocytes, and we abbreviate them RBCs. You'll see white blood cells, which are also known as leukocytes, which we call them WBCs, and then platelets. Okay, so we have way more red blood cells than we have leukocytes and platelets. The platelets are just really, really small. So the erythrocytes, or the red blood cells, are this red layer here. The white blood cells and the platelets are this thin they call it Buffy coat. The white layer, it's really thin. And then on the top, okay, that's where the plasma is going to be. So the plasma, it's, sometimes they describe it as straw colored. So like the color of hay or straw. So it's kind of a clear, light yellow fluid. So it should be clear. You should be able to see through it. Okay, so connective tissue. Okay, so here's something. This is just to get you started, and we'll do this in lab. This is a hematocrit. So a hematocrit is a test that you do to determine how many red blood cells you have. So you can figure out your percentage of red blood cells from a hematocrit, and that's actually used to determine whether you're anemic or not. So they do that like when you're pregnant. If you go have WIC, which is women, infants, and children, they want to make sure that you have enough like iron in your diet and you're eating healthy. So they always do a hematocrit when you go there. So how a hematocrit's done. Okay, so first thing, this is messed up. You can see the little glitches. First thing you do, it just takes an itty bitty amount of blood. It doesn't take much at all. So you do it like a finger stick, or they're doing a whole draw. Okay, we can use a little tiny capillary tube. So you do a finger stick and you suck the blood up into it. The blood just kind of gets pulled up in by capillary action. Okay, so you collect the blood. You spin it in the centrifuge, and the centrifuge, all it does is it spins like this, and it's going to separate things out by size faster. Okay, so separates the components of the blood by mass, and then what you do is you're going to take a ruler and you measure it. Okay, so you measure it in millimeters. Okay, so you just measure it with the ruler. And then what you do is you take, let's say this is 2, and the total distance, so this is 0, this is 2, and let's say this is 5. Okay, so what you would do is you take the distance of your red blood cells, which is 2, and the total height, which is 5, right? And the units are the same, so they're going to cancel out. And multiply it by 100, and that's going to give you, I don't know what it gives you, because I didn't pick a nice, 2 divided by 5 would be, what, 40%? I'm trying to mess with my phone to get to my calculator. It's pretty bad, huh? 2 divided by 5 is 40, yep. So that's 40%. And that's the hematocrit. So out of this person's total volume of blood, 40% of it is red blood cells. Okay, we'll do this in lab. I wanted you to see this so you could see the different components of the blood. So they'll separate out. So you'll see white blood cells up here in the Buffy coat, the red blood cells, and then the plasma. This one, they actually said is 45 when they measured it. So I didn't measure it for real. I faked those numbers, and obviously they weren't right. So, but we'll do it in lab. You'll get to see it. Okay, now, when we talk about formed elements, what we're talking about are the cells in blood. The first kind we've already hit on is red blood cells, so erythrocytes. The second kind are platelets. Okay, so platelets are cell fragments, and they come from this specialized cell, and they're made in the bone marrow, but then they get released into the blood. So we'll talk more about them. They're important in clotting. Okay, now the third type, which actually has 5 subtypes under it, are leukocytes, or white blood cells. Okay, so white blood cells are divided into 2 broad categories. Okay, so A is granulocytes, and B is agranulocytes. Okay, so when you look at these under a microscope, granulocytes have cytoplasmic granules in them that you can actually see. So the cell, the cytoplasm, looks grainy. Okay, so granulocytes have 3 subtypes. Okay, so there are 3 kinds of granulocytes. There are neutrophils, eosinophils, and basophils. And in my mind, to remember this, I always say Ben and Phil. So you remember the Phil part too. Loves Granny. So Ben loves Granny is how I remember those. Okay, then agranulocytes, their cytoplasm doesn't have those granules. Okay, so they're clear. So those kind are lymphocytes and monocytes. Okay, so if we go back and count the 7, so we go 1, 2, 3, 4, 5, 6, 7. Okay, now we're going to spend a lot of time on these in lab. We have some pictures, some flashcards, so you're going to be able to recognize them if you look in a microscope. This picture, I think it's a scanning electron micrograph, and so it's really, really magnified, and it's colorized, so they're making it look all pretty. But red blood cells look red. This is a white. The platelets are these little fragments. Okay, now, this is messed up. I entered it into the iPad. It looks weird, sorry. Okay, so first thing we're going to talk about is plasma. Okay, so you probably have heard about plasma. So the difference between plasma and serum. Okay, plasma is, it's all the liquid of the blood. Okay, so that's the extracellular fluid. When you have serum, what you've done is you've removed the clotting proteins and the cells, the formed elements. Okay, so that's serum. So serum's a little bit different. It's just plasma without the clotting proteins. So maybe I should write that here. Serum equals plasma minus clotting proteins. Okay, now plasma and serum, they're mainly water. Okay, so they're like 92% water. Besides water, they also have other kinds of plasma proteins in them. Okay, so the plasma proteins are actually really super important for the function of blood. So the first kind is albumin. Okay, so albumins, I don't know if you've heard of it or not. You probably haven't, but there's, like, you can have egg albumin, you can have cow albumin. It's just protein, and what it does is it contributes. It's a specific protein. Okay, so it's you can buy it for working in a molecular biology lab. It looks white and fluffy if you dry it out. What it's going to do is it's going to help you regulate the viscosity and the osmolarity of the blood. Okay, so how concentrated the blood is has a lot to do with the albumin. Okay, so it's going to influence blood pressure. It'll influence how the blood flows. It'll help influence whether you have edema or not. Okay, now globulins are another kind of plasma protein, and there's 3 kinds of them. Okay, so there's alpha globulins. They transport lipids and metal ions. There's beta globulins. They transport iron and lipids. And then the big one that we're more worried about are gamma globulins. Okay, and gamma globulins are actually antibodies. Okay, so the antibodies are going to be part of your immune system and are going to help protect you from pathogens. Okay, now the third plasma protein is fibrinogen, and fibrinogen is the precursor to fibrin. And fibrin is going to be the structure that makes a blood clot. Okay, so fibers in a blood clot, and we're going to talk about that more. So it's a key component of blood clots. So all of these, well, albumins and fibrinogen, okay, and alpha globulins and beta globulins, Those are all made in the liver. Okay, so the liver is important for your blood production. Okay, now, the non-protein components of plasma, okay, so only 1% of the blood. Other things that are found in there, okay, are electrolytes, nutrients, respiratory gases, okay, so carbon dioxide and oxygen, waste, and that's it. Okay, so that's actually a really small amount of the blood. So the main thing is water, then we have protein, and then we have these non-protein components. Okay, now, more properties of blood, and this is messed up, because I imported this weird. Okay, so more properties of blood. So I want you to think about osmolarity again. Okay, so osmolarity, how it's kind of defined is the total molarity of dissolved particles that cannot pass through the blood vessel wall. So basically what it is, it's the amount of solids that are in the blood, okay? And they're going to contribute to the concentration of the blood and how liquids can move, fluid moves. Okay, so here's the problem. Okay, and this is supposed to look like this. Okay, so here's our beaker. Okay, here's the cell in the beaker. Okay, and the cell, if you look at it, has 1% sodium chloride and 5% protein. Okay, out of this cell, the fluid in the beaker, okay, what's around here, is 1% sodium chloride and 3% protein. Okay, so basically we're talking about the osmolarity. Okay, so these solids can't exit the cell, so they're trapped in here. Okay, so the only thing that can move is the water. So which way is the water going to move? So if I look at this, I would say, oops, there's my phone. I would say this is 6% solids, right? And this is 4%. So that means I have 94% water out here and 96% water out here. So that means, okay, so this is referring to, that means that the water is going to flow from outside the cell into the cell. Okay, so the water is always going to follow the solids. Okay, so different things going on in our body can alter the amount of protein that's in there, and that's going to affect how the water moves. Okay, so here's another picture. So how can osmolarity of blood affect the body? Okay, so let's say this is my blood vessel. Okay, and then this is my tissue with my cells. Okay, so if you look here, okay, so this is 1% and 3%. Okay, so water is going to move. So let's say this person is dehydrated. Okay, so water is going to move from out here into the bloodstream. Okay, so what's going to happen to your blood volume? Blood volume would increase, and then what happens to your blood pressure? If your blood volume increases, your blood pressure would go up. Okay, so think about if you eat something really salty, okay? So like if you're drinking a bunch of Coke and eating a bunch of pretzels or chips or something, okay, and then your concentration of your blood goes up, okay? So you have increased sodium in there. The fluid is going to move from the tissue into the blood. It's going to make your blood volume go up, and it'll make your blood pressure go up. Okay, so I have that written out here. So if the osmolarity is too high, blood absorbs too much water, which increases the blood volume, and in turn is going to increase the blood pressure. Okay, now, if the osmolarity is too low, okay, so something's going on and these numbers are lower, okay, the water's going to go to the tissue, so it would go the other way. And that's going to cause edema to occur. It has liver disease. Okay, now, what could change the osmolarity of the blood? Okay, so if somebody's starving, they don't have enough protein. Okay, so extreme starvation, so they have a decrease in protein. So I have hypoproteinemia. Okay, so hypo means they have reduced protein. They're going to have edema. Okay, if you have liver or kidney disease, okay, so the liver is supposed to be making those proteins. If it's not functioning, you're not going to get it. You're going to have edema. Okay, so severe burn patients, they lose a lot of protein. Okay, so I can never say this right. I'm not sure how you're supposed to say it. Kwashiorkor. It's for children with severe protein deficiency. So sometimes it occurs in developing countries where kids are, if they're weaned, their diet is not very good. Okay, so they're fed on cereals if they're weaned and they don't have enough protein in their diet. And what happens is they end up with this distended belly because there's fluid accumulating in there. So the muscle tone is bad because they're not developing muscles. Sometimes you see, like I have a friend, her brother had Crohn's disease. And I mean, he's had it forever. So he wasn't absorbing his nutrients well enough. Okay, so he kind of looked like this. And that's the end there.